The Front Range is home to many of the remaining glaciers in Colorado, several of which lie within the Indian Peaks Wilderness area. Due to climate change, these glaciers have seen a rapid decline in surface area and thickness throughout the 20th and 21st centuries, but are currently stable, supplied by large amounts of wind-blown snow. This snow not only maintains the mass balance of the glaciers, it also deposits dust, organisms, and dead organic matter. These deposits create unique and isolated microbial communities on the glacier, known as cryoconite. The dark color of sediment and dust on the glacier absorbs energy from the sun and melts small microenvironments of liquid water, allowing microorganisms to survive in these seemingly inhospitable conditions. Our lab has studied cryoconite communities around the world, including in remote valleys of Antarctica and Svalbard. However, the cryoconite on glaciers in our own backyard has never been studied.
As these glaciers melt, they expose land which has not seen the light of day since before the last little ice age. This provides us a rare opportunity to examine alpine soil development in real time after glacial recession, helping us to understand the role glacial cryoconite microbes play in alpine soil development and succession. Hardy microbes which can survive extreme conditions colonize this land and develop it into soils, allowing other microorganisms and plants to start growing there, which changes the dynamics of the local ecosystems. Our lab studies these sorts of soil development processes in the forefields of glaciers in Svalbard and Peru, and we were curious to see how they compare to the development of new soils in the Colorado Rockies. Currently, there is no published scientific literature studying these unique and isolated glacial environments in the Front Range, so we set out to sample cryoconite and forefield soils from Arikari and Isabel glaciers. We sampled areas that have been previously covered by glaciers, or are predominantly snow-covered for the majority of the year, and moved away from those locations until we reached areas with developed soil that could host plants and mosses, to understand how glacial proximity changes microbial community composition.
We are currently in the process of extracting the DNA for all of our samples, and we will have results to share from this field season coming spring of 2025. This research was made possible thanks to a generous contribution from the Indian Peaks Wilderness Alliance, and in collaboration with the Mountain Research Station, and the University of Colorado Department of Ecology and Evolutionary Biology.